Good afternoon, everyone. Welcome to the AI Plus Seminar Series from Algorithms to Impacts. And this is all part of a UQ AI Collaboratory initiative. And isn't it great to be back face to face? So I know many of you have really been dying for opportunities to come along to these sorts of events in person and interact and network. So welcome everyone who's here today, and also welcome to those online. So firstly, I'd like to acknowledge the traditional owners of the land and their custodianship on the lands on which we meet today. This AI Plus Seminar Series, it's all about promoting transdisciplinary AI research and collaboration, and really trying to foster networks and collaborative work across the broader UQ community. So it really doesn't matter what school or faculty you're in. This is an area of interest for a lot of our researchers and academics here at UQ, and really spanning areas from agriculture, health, and energy. So far in this special AI Plus series on agriculture, we've had several sessions. So here you can see we kicked the session off with a talk from Scott Chapman, followed by Andreas Potgieter, and now today we have two other leading experts in the field to share their experiences. And so here you can see them on screen. Firstly, I'll introduce myself as the chair today. I'm a plant breeder geneticist based at Coffee here at UQ, and my new role in Coffee is leading the theme of predictive agriculture. And I think this seminar series and initiative really pairs nicely with the objectives of what we're trying to do with the theme in this space. So again, really trying to foster collaboration. And so today I think is a great opportunity to hear a bit about from research from Coffee, but also, I think we can all network and interact. After the talks today, we're going to have some catering outside. So please stay around for that. And so I think it's really some exciting opportunities to be collaborating across, faculties, in particular, ITEE, SAFs, and Coffee in this agriculture space for the future. So to kick things off, I'll first introduce both of our speakers. So I'll start with Professor Mark Cooper, who is the chair of prediction-based crop improvement here at UQ and a global leader in quantitative genetics and plant breeding. His work involves integrating genomic prediction and crop growth models into an end-to-end framework. Might pick up my little sheet. For crop improvement. Professor Cooper is also the Deputy Director of the ARC Centre of Excellence for Plant Success in Nature in Agriculture. Mark started his career at UQ and then moved to industry, working at DuPont Pioneer on drought adaptation in one of the largest maize breeding programs in the world, where he played a leading role in developing Aquamax hybrids that presently cover millions of hectares globally. Now he's back at UQ, building on the critical mass of predictive agriculture expertise at UQ. Secondly, I'd like to introduce Professor Ben Hayes, who has extensive research experience in genetic improvement of livestock, crop, pasture, and aquaculture species, with a focus on integration of genomic information into breeding programs. He is the author of more than 300 journal papers, contributing to statistical methodology for genomic, microbiome, and metagenomic profile predictions, quantitative genetics, including knowledge of genetic mechanisms underlying complex traits, and development of bioinformatic pipelines for sequence analyses. I think impressively, he was one of the co-inventors of genomic selection, and that paper, which he published with his colleagues, Mayerson and Goddard, has been published almost 5,000 times since 2001. And as a result of all this breakthrough research he's been doing, he's been named as a Thomson Reuters highly cited researcher almost every year since 2015, which is pretty impressive. So first, to kick things off, I'd like to welcome Ben. Is that right? That's okay. So we've got it on the webcam view, so if you're looking to.
Yep. Hi all. First of all, thanks very much for the invitation to talk today. As you'll see at the end of the talk, we really are looking for collaboration. As Lee said, my area of expertise is quantitative genetics, definitely not AI. We're looking to use AI as a tool in breeding, and I'd very much like your help on that. But before we get to that, I'll take you through some of the things we have been doing. So just a very brief outline of the talk. I'm just going to have a few brief slides on what selective breeding actually is and what it's achieved. Then go into genomic selection, how it works, entertain the possibility of machine learning for genomic selection. Then talk about another approach, I guess, to breeding, which is an AI-guided breeding tool called FastStack, and I'll explain what that is. And then last of all, finish up with a request for help from you. So selective breeding is a really powerful science, and I'm about to show you a few examples to hopefully convince you of that. But just to give a bit of background, what is selective breeding? What you're attempting to do in a breeding program is to select those parents that when you breed them and they leave behind progeny, they have the highest genetic merit, which they pass on to the next generation. And if you do that generation after generation, you can make quite considerable gains. In our selective breeding since about the 1950s, we've been using a tool called best linear unbiased prediction. And this is basically a framework to separate out the environmental effects that might contribute to an animal's performance or phenotype from the genetic effects. So we use this tool BLUP, or best linear unbiased prediction. It combines things like the animal or plant varieties' own records for yield, milk production, whatever your trait target is, with information like the pedigree, who's related to who in the breeding program. And that lets you tease apart those environmental effects from the environmental effects. If you've ever used ridge regression, BLUP is a form of ridge regression that takes into account the relationship between individuals. The BLUP theory was actually developed based on the infinitesimal model. And if you're aware of the history of genetics, there was this long-term, I guess, disagreement between the Mendelians, derived from the priest Gregor Mendel, who observed that some effects, genetic effects, occurred in a Mendelian fashion, so large effects that were passed down, compared to the, let's call them complex trait geneticists. And Fisher very nicely reconciled those two opposing ideas, I guess, with the infinitesimal model, which says the complex trait variation we observe, so variation in traits like milk yield, like disease resistance, height in humans, any other trait you can think of, is the result of an infinite number of genes of infinitely small effect underpinning those traits. So all those genes operate in a Mendelian fashion. There's just an infinite number of them, and they all contribute to the trait variation. It sounds really weird, but the interesting thing is the better our genomic tools get and the bigger our data set gets, the more it looks like traits like human height, milk yield, and so on are pretty close to this infinitesimal model. They're not exactly the infinitesimal model, that's impossible, but there is a very large number of genes or mutations affecting the traits we're interested in. And I'll come back to that throughout this talk. Okay, just to convince you that selective breeding is a powerful science, as an example, through selecting chickens for faster growth, using exactly the technology I've just talked about, estimated breeding values, the time to market weight for meat chickens has decreased from 16 to 5 weeks in 30 years. So the picture there is of 2 chickens, identical stage in life. One is from the selected commercial lines. One is from a historic sort of backyard collection, but the same original germplasm. You can just see what a tremendous difference there is. And in terms of efficiency of utilising feed to produce protein to feed us, it also comes across in what's happened in salmon breeding programs. Feed conversion efficiency has been improved by 140% in salmon in just 7 generations. So the amount of food they need to put on a kilo of salmon fillet has just shrunk enormously. And in terms of feeding us with reduced resources, that's pretty important and pretty impressive. Same thing has happened in plants. This is a very nice paper that was published, I think Mark is actually co-author, showing the genetic trend or the improvement in yield in corn over a span of about 70 or 80 years. And you can see every year the yield we get is just going up and up and up. And this is dissecting out the genetic component from things like improved fertilizers and pesticides and so on. So it's about 77 kilos extra per hectare per year. And if you think about having to feed a world population that's growing on less resources, this is a pretty powerful tool to do that. Okay, so that's how selective breeding was operating really up to about 2010 or a little bit earlier. And then we had this amazing new tool that we could incorporate in our selection programs. And that was information at the DNA level of variation between individuals. And that came through sequencing large numbers of individuals of the same species. And the variation, the main source of variation, not the only source, but the main source I'm talking about is single nucleotide polymorphisms or base pair differences between individuals. So if you sequence 2 cows, just if you sequence 2 humans, you'll get 3 billion ACTs and Gs. And at a very small proportion of those ACTs and Gs, you'll get differences in the base pairs. And here 1 cow carries the G allele at a SNP, the other a C allele. I say a very small proportion, but because there's 3 billion bases, there's actually a huge amount of variation to work with. And in our cattle sequencing collections, we now have about 70 million SNP variants that we know about. And in humans, from the 1,000 Human Genomes Project, it's about the same order of magnitude. The great advantage of this SNP information is it's cheap and easy to get. So from a tail hair or a hair off your head, as long as you get the bowl on the end, you can extract out the DNA and run it across these small glass slides, which are called SNP chips or SNP arrays. So they'll assay SNP variation at say 50,000 or even up to 700,000 points across the genome. Okay, so that's terrific. We can get this information on our cattle, on people, on plants, fairly cheaply and easily now. What do we actually do with it? So how do we translate this genomic information into estimates of genetic merit of individuals for breeding, estimated breeding values? So this is what I'm about to describe to you is genomic selection. So this is the technology we developed back in 2001. So what you require, first of all, is a very large reference population. So this is a group of animals or plants or even humans with traits measured, with something like height measured or disease measured. If it's livestock, it might be growth or milk yield or disease resistance. At the same time, you also have phenotypes on those individuals. Genotypes. So you have their SNPs, their DNA run across the SNP chips, their SNPs characterized. And this is a cartoon of that SNP information. So this is a particular chromosome. The M's are the DNA markers, the SNPs. The Q's are mutations affecting the trait that might lay in between the SNPs or may not. So what you do in genomic prediction is you work out the effect of the SNPs on the trait or associated with the trait. So say it's growth in these pigs, you might see that all the pigs that carry the AA genotype, when you compare them to the AT genotype, all the pigs that carry the AT genotype, so the heterozygous individuals, the ones that have got a T allele, they have an extra 0.02 grams of growth. And the ones that get a TT genotype, they have an extra 0.04 grams of growth. So then the weight for that particular SNP would be 0.02. And what's actually happening there is the SNP is very closely linked or tracking an underlying mutation that, or a gene that is causing differences between animals in the growth rate. The key thing about genomic selection is it was developed because we and others realized that these complex traits like growth, like yield and so on, they were underpinned by many, many mutations across the genome. It just wasn't one gene that was giving you big differences. It was lots and lots of genes, all of small effect. So you have to have these many SNPs across the genome and you put weights on all the different SNPs there. We'll go to the mathematics in a little bit on how you do that. But once you do that, if you can do that, you have a prediction equation and then you're in a really good position because it means you can come to a group of very young pigs or very young animals or seedlings, and then you can genotype them, run their DNA across the SNP arrays, run their genotypes through the prediction equation and come up with their estimated genetic merit, their prediction of their value for a particular trait, their genetic value. In humans, of course, when you're using this technology, you stop there. You might predict someone's disease or future attributes and so on. In livestock and crops, we can go to the next step, which is selecting out of those individuals which ones to breed from. And if we've got a large enough reference population, done a good job on our prediction equation, that estimate of genetic merit might be very accurate, and therefore we can make very good selection decisions. So that is genomic selection in a cartoon. You may be sitting there thinking, how on earth do you derive that prediction equation? And it is a big challenge because typically you've got a P much greater than N problem. You've got many, many more SNPs than you have observations. So you might have 2,000 records and 50,000 SNP effects, SNPs. Or if you go to whole genome sequence, it gets even worse. We have reasonable data sets, 30,000 animals now, but you can have 45 million or something SNPs that you're trying to predict the effects of. So how do you do that? So our friend from the 1950s, Blup, turns out that model or probably more precisely in this case, ridge regression, turns out to be quite a good method for predicting SNP effects from such data. And the reason you can do it is in the BLUP model, you effectively take in a prior assumption that the SNP effects are all derived from a normal distribution, and you're really assuming that you have many mutations of small effect. In fact, it's very, very similar to the infinitesimal model. So we do use a BLUP model quite routinely to estimate SNP effects. And I guess the interesting thing is, after 10 or more years of doing this BLUP model turns out to be quite robust across species and a range of traits. And it tends to be what people use in routine breeding programs. And just to show you the impact of this, we implemented this technology in the Australian dairy industry in 2010. So we actually started running genomic predictions on all the young dairy bulls that were being born, and the breeding companies worked with that to select out the best ones from breeding. And this is the impact that it's made on the Australian dairy industry. So the trait I've got up here is fertility. And fertility in the Australian dairy herd, as for around the world, was deteriorating very rapidly. If you go back in time, if you just focus on the blue line, you can see this negative trend for fertility. So the y-axis, you can think of that as it's how easy cows are to get pregnant. And that was starting to drop off a cliff. And that was because they were selecting really quickly for milk production, and fertility is a negatively correlated trait with milk production. So it was going backwards. We implemented genomic selection in about 2010 for milk production, and you can see fertility really started to improve again because we had a heavy weight for fertility. So our genomic selection for fertility really worked. In the US, they used our method, but they're actually a bit earlier in getting onto it. So they started for milk production in 2008. And if you just look at the blue line, on the y-axis is effectively milk production. The x-axis is time, and you can see the milk production has improved enormously. They also selected for improved fertility, and they've improved that as well. Just really quickly, since this title of this talk was about the breeder's equation, the breeder's equation tells you why this actually happened. So this is the breeder's equation. Delta G is the genetic gain. So it's what you're seeing on the y-axis. It says the genetic gain you observe is the intensity of selection. So how many bulls you select out of all the bulls you test. R is how accurately you do that, the correlation with the true genetic value. Sigma G is the genetic diversity you have. And you divide that by L, and that's the generation length. The key thing that happened in the dairy industry was I and R stayed about the same. R was actually a little bit reduced compared to what they were doing before, the accuracy of selection. But they were able to select bull calves basically at birth and use them for breeding at about 12 months when they were reproductively able, rather than wait for 5 years to use the same animals, which is when they started to have good information on milk yield, because you could see how their daughters were performing. So basically, the dairy industry cut their breeding cycle from 5 to 6 years down to 1 year. So if you cut L down from 5 to 1, even if you have a little bit less accuracy, you get this acceleration in genetic gain. So that's where the breeder's equation comes in. So big payoff there. Okay, back to deriving the prediction equation and towards machine learning as well. Deriving this prediction equation is a challenge, as I said. BLUP gives you a robust answer. Is it the best answer? Almost certainly not. Quite a bit of my career has been taken up with Bayesian approaches to get more accurate genomic predictions. And in the Bayesian approaches, it's a nice framework for different assumptions about the effects of these mutations on the trait. For example, you might want to assume that in some regions of the genome, it doesn't have any effect on the trait. There's no gene or mutation there affecting the trait, whereas in others, there's a small or moderate to large effect, especially when you've got sequence data to play with. So we've shown those Bayesian approaches can have advantages, maybe 10 to 20%. No, that's exaggerating a bit. Let's say 5 to 10% extra accuracy. Best case scenario, 20%. More typically 5 to 10, but not a huge advantage over BLUP. So then we started to look around and ask the question, could we apply some of these machine learning approaches to this problem of genomic prediction and increase our accuracy? Because if we can increase our accuracy, the breeder's equation says you'll get faster genetic gain. So there's a little bit of a history here. People started applying machine learning type approaches to genomic prediction probably in the last 7 years. And to tell the truth, you can look up the literature, there hasn't been huge gains. For some traits, maybe 2%, other traits, not as good as BLUP. And we can discuss reasons why that is. So we were still interested in doing this, and we thought, why don't we throw these machine learning approaches at the hairiest, ugliest, worst genome imaginable, the worst species that we've ever come across for genomic selection. Any guesses on what that might be? Yeah, sugarcane. The plant breeders know it. So sugarcane, if you've ever looked at the genome, you just look at it and want to look away because it's got multiple, well, first of all, it's a hybrid of 2 species. The species had multiple genome duplications, and there's since been more duplications again. So it's anything, Craig can tell me more accurately, but from 8 copies of the genome to 10, and it differs in different strains and so on. It's just a really messy genome. And it makes things like banana, which are triploid, and potato, which are tetraploid, look really clean and nice. We're diploid, of course. We got one from mum and one from dad. Very, very simple. Okay, so we thought this would be a good species to test it out on. We also knew that from work a PhD student of ours, Seema Yadav, had done previously, that there was a lot of non-additive genetic variation in sugarcane. Unlike cattle, where we don't see that much non-additive variation, in sugarcane, there seems to be a lot of dominance and a lot of epistatic variation. Whether it is truly epistatic by the classic definition, we don't know, but certainly when you fit epistatic and dominance models, they pick up a lot of variation. And this is just a graph from Seema's paper. If you just look at the last column in each block, the first column is cane yield, total cane harvested. The second one is commercial cane sugar. And then you've got fibre, 3 key traits in sugarcane. The last bar shows you when you put the full model in the phenotype, cane yield, this much is error, environmental variation. This much is epistatic, the green. The little blue bit is dominance, and the next bit is additive. So certainly for cane yield, there's lots of non-additive variation going on there. So we thought this wasn't a bad species to throw machine learning at. The other nice thing about sugarcane, there is something nice about it, is that it's clonally propagated. So when you find a variety or line that has the best sort of stack of additive variation, non-additive variation, epistatic variation, you can clone it almost infinitely just by cutting the stalk and planting out the stalk. And that's how genetic improvement is actually delivered to sugarcane farmers as these sort of stakes. So if you find something that stacks all this non-additive variation up, you can actually use it for the farmers. Okay, now I'm getting on to a PhD student of ours, Chen Song, who's done a really good job, I think, of looking at different machine learning approaches for prediction. This is for discussion at the end as well. He's used things like convoluted neural nets, random forest to assess the predictions against our standard BLUP models to see how they perform. Okay, just to give you insight into the data we're actually using here is almost 4,000 clones, which have SNP chip data, 26,000 SNP, we think across the genome, though it's hard to know because it's so messy. And they've got these traits, yield, fiber content, and commercial cane sugar. And what Chen Song has done in his work is he's dropped out the final year of data, so clones that did actually have all that data, predicted using the machine learning as well as the BLUP, what those clones should be doing for those 3 traits. So he gets genomic predictions and then he correlates those 2 things. So actual performance and the prediction. And this is the result he gets. So I should tell you that cane yield is a very hard trait to predict in sugarcane. Things like fiber have a relatively high accuracy of prediction. And what Chen Song observed is that for cane yield, the convoluted neural net had the highest accuracy of prediction by a little bit of a margin compared to random forest and then all of these variants of our BLUP approach, that infinite model that assumes the infinitesimal model. For the other traits, the BLUP and the machine learning approaches pretty much did equally well. If you remember a little bit of information on the underlying architecture of the traits, cane yield, this one here, was the one with the highest non-additive contribution to variation, whereas these traits had quite a bit less. So what we think's going on is the machine learning is able to pick up some of that non-additive variation and use it in the prediction. Okay, that's for prediction. The other thing Chen Song's done quite nicely is look at, I'm not sure if you call them parameters, but inputs into the convoluted neural nets and so on, like the number of neuronal layers and so on, and looked at how that affects accuracy. And that, just in very brief summary, it seemed to point at that for things like cane yield that had a lot of non-additive effects, you needed more complexity in those models, whereas the other traits, you could get away with reasonably simple models. Okay, I just want to finish up now, almost done, with another potential use of, I'm very loosely going to call it AI. Some people might object to that, but very loosely call it AI approaches that we can use to really have a new look or a new approach to breeding. And the overall idea, I guess, is with our genomic data, what we can do is we can stop thinking of a breeding program as being a collection of individual lines or individual animals that we're going to breed from. So in a standard program, you calculate breeding values for each of those lines or animals and then breed from them. In this new approach that we think might be worthwhile, you consider the population that you've got as a population of chromosome chunks, bits of chromosome, all with their own breeding value that for the trait you're targeting, like yield or resistance, that you can stack together to create an ultimate variety or line. Okay, so you stop thinking about a line. A line might have one particular chromosome chunk that's really good for a trait, and the rest might be really bad, and that would come out as a low genomic breeding value. So you would never consider that line. But in this approach, you would see that chromosome segment and you would say, hey, I actually want to use that line in the breeding program to then start to stack it with other chromosome segments in other lines to create this ultimate variety. And myself and my colleagues have done work where we've looked at chickpea populations. If you take the best chromosome segments in the real population, when you work out those chromosome segments using the genomic data and you stack them together, you get ultimate varieties that might have double the yield of any variety that exists today. Just tells you how far you could go with your breeding. The twist we're going to use here is we can define that ultimate variety, and then we can say to an AI system, work out the system of crossings, the crossings between the lines or individuals to stack up that ultimate variety in the shortest time possible. And we can combine that with technologies like Leah's developed, called speed breeding, where you can breed crops under lights to crank through 7 generations in a year instead of having to wait 7 years to get through those generations. In cattle, the equivalent is things like IVF, in vitro fertilization. So these technologies combine. It's not really AI, it's really an evolutionary algorithm. I guess it falls loosely in this class. That we've used for this is a genetic algorithm. If you're familiar with genetic algorithms, this is an optimization method where you represent the problem as a series of chromosomes that you can recombine and have mutations and so on. So it seems to work for geneticists quite well. And we've used this algorithm, but it could be replaced by others. Just to finish up on what we're doing, this work is ongoing, but a very talented undergraduate student that's working with us, Kira Villas, has coded this up, and she runs these breeding programs forward in a digital twin, a simulation of the breeding program, and looks to see where you could get to after multiple generations. Here she's starting with a real wheat breeding program, a wheat germplasm collection, and she asked the question, how far can we go in terms of yield, and how far can we go in terms of maintaining diversity in that program? Because genetic diversity, if you remember your breeder's equation, that's your future genetic gains. That's your investment in the future. If you erode that, you're buggering up the future, particularly if you want to start selecting for new traits. So the A there is just the germplasm collection. Every row and column is an individual. It just shows how related they are. So the darker it is, the more related they are. The next graph along is what the genetic algorithm is actually choosing for the parents. So this is a principal component plot. So it's pulling apart the potential parents on the genetic diversity. And then the size of the circles is how many offspring you're breeding from each combination. And what you can see is, if you look hard enough, with the old style selection, truncation selection, you tend to breed a lot of offspring from one cross because it's a very good cross, and your intuition tells you, and the program tells you, breed a lot of offspring from that cross. The genetic algorithm says, no, I actually want to get these chromosome segments from a number of different parents and stack them together. I'm going to have a more balanced crossing strategy across the parents. This is very close to the final slide, but in this particular example, when we ran it, the genetic gain after 25 generations from the GA genetic algorithm and the traditional truncation selection, where you just choose the best every generation, was actually quite similar. If you just look at this bar here, this is when you breed 100 progeny from each cross. This is for the GA, and this is for truncation selection. So quite similar there. The really big impact is on your future gains, your future genetic diversity or your genetic diversity. And this is the amount of diversity you have in your breeding program after 25 generations. So with the GA, you still have a reasonable amount. With the truncation selection, it's gone. You've basically used up all your genetic diversity by that point. So you made good genetic gains for 25 generations, but then you plateau because you can't go any further. And that does happen in some real breeding programs. But your genetic algorithm has stored up genetic diversity. And it's because it's used many different parents to pull those different segments from. That's just one example. Okay, so to finish up, I think machine learning does have some application in this task of predicting which parents are best to use for the next generation, particularly when you've got highly complex genomes and extensive non-additive variation. And particularly when you can take advantage of that non-additive variation through things like cloning, clonal propagation, and so on. This approach I've just introduced called fast stacking, where you take these chromosome segments and stack them together, it seems to have some advantages, particularly in maintaining genetic diversity. So it's an interesting approach to animal breeding to look at. Now, as I've been talking, those of you familiar with AI approaches and machine learning and so on, I've probably been thinking, oh, there's a much better method for doing that. We could use this, this, and this. So that's exactly the purpose of presenting to you, is to get your feedback on what we could be doing better. We're looking to collaborate, and any ideas you have would be very appreciated. Just finally, this is an important problem. It's a global problem. Genomic selection is being rolled out across the globe for many, many species. This is just an example of a part of a genomic breeding program for cassava in Africa, which is a food security crop. And these cow and a buffalo are part of a genomic selection program in India, selecting for higher milk yield for Indian children. So it really does have global implications if you're looking for an area that's going to have quite an impact. So thank you, and thank you all the people that contributed to this too, Chen Song and Kira in particular. Okay.
Okay, well, we'll just hand over to Mark.
Thanks very much. Really glad to be here. I'll jump straight into it, and it's really nice to follow on from Ben. He introduced a lot of the concepts that I'll cover. So given that I had some idea of some of the topics that Ben would cover, I thought I'd sort of discuss this from the perspective of being a commercial maize breeder. So I was a commercial maize breeder for a couple of decades, responsible for developing technologies, but putting those into practice. And ultimately, what we were doing was developing seed that is then sold to farmers, ultimately to be adopted and grown sort of in a lot of areas. I lived through many mergers and acquisitions as companies changed, starting off with Pioneer Hybrid, eventually ending up as part of Corteva, which still exists today. And Corteva are one of the partners of the ARC Center of Excellence for Plant Success in Nature and Agriculture that was introduced previously. And so I'm going to talk about a lot of published work that I was involved in while I was involved in breeding there. And I'll sort of revisit some ideas around the breeder's equation that Ben has already introduced. So let's see how we go. So the way I thought I'd do this is, before jumping into an outline, is just give you my perspective as an actual commercial breeder of what is an actual maize breeding program. What is a plant breeding program? And so as a plant breeder, obviously we grow a lot of plants. And so some of you may be really familiar with how a breeding program looks, but I'll sort of try and give you a bit of an introduction. So it's very easy to go through the mechanistic aspects of what a breeding program is and describe it. But the message that I want to give you is that it's a really strongly biologically inspired search algorithm that's exploring genetic space in combination with an understanding of a target population of environments that are the agricultural environments that sit inside a series of ecophysiological environments around the globe. So that's actually what we're trying to explore. The unique thing that a breeder does that everybody else doesn't do is they manage and improve germplasm. So germplasm is a resource that you actually create and actually use it to solve problems in combination with other technologies. And just to give you a bit of an impression of the search algorithm aspect and the exploration, this is sort of a history of germplasm that was improved from founders, where the lines indicate parent to children trajectories, and the germplasm trajectories through genetic space that were created over somewhere around about 70 years is represented here of breeding, which is multiple cycles of breeding programs. So in here, you've got the founders that go out to sort of much newer germplasm on both sides of the hybrids that we create. So we created these heterotic groups that are then combined to produce these commercial hybrids. And so as a breeder, what I'm doing is I'm taking the genetic resources, intercrossing those to create a whole new series of recombinants to explore other features of this genetic space using the technologies that Ben talked about and a lot of biological knowledge that determines which crosses to make and then which individuals to select out of those crosses. So that's sort of conceptually what we're actually doing. As a large-scale multinational commercial breeder that operated around the world, the germplasm's there. We have an operational breeding program. So if you look at this little cartoon on the side here, it sort of represents a breeding program. These two boxes up here represent this germplasm on the male and female side of the hybrid. These are the crosses that I highlighted before. Then we go into some sort of testing to sort out all these individuals that we've actually created and refer to these as multi-environment trials. So here's an example of all the different stages of my breeding program when I was a maize breeder at one of the research stations that I operated at. So every stage of this breeding program is actually running simultaneously at this location. So basically I'm running probably about 20 breeding programs all at once on the male and female side at all different stages, making decisions all the time. So a lot of these technologies and what Ben was talking about, a large part of what you have to do is work out how to operationalize this, where you're sorting out millions of individuals simultaneously for all these different target environments. You're doing this in a very small sample of environments to try and represent expectations for this large target population environments. And so here's an example of how this is scaled up for one of the experiments I was involved in. So this is the US. I'll come back to this a little bit later. And each point is an experiment on the ground in a farmer's field, sorting out a whole series of hybrids that we've created for one of the stages of the breeding program. But there's multiple of these testing stages going on simultaneously. And we've done a whole lot of environmental characterization work to sort out from all of these experiments, which ones, the ones that are highlighted in red, where we've identified that water limitation was a factor that was driving the expression of the variation we were studying. So we're understanding the germplasm. We're measuring a lot of traits, as Ben talked about. We're also characterizing our environments so that we can go from these experiments to make inferences into a larger target population environments. And simultaneously, all these individuals are genotyped, as Ben described. So whenever I get a bag of seed back, here's your new progeny, it's already genotyped. So the type of prediction that Ben was talking about, I could do that even before I got the bag of seed. So I was running all these predictions to decide out of the new million progeny I'd created each year, which ones would I actually take into my nurseries and into the start of my next cycle of a breeding program. And then the other thing that we're spending a lot of time doing is trying to understand the genetic architecture of these traits. Ben introduced you to the basic principles of the infinitesimal model. Here is a cartoon of this infinitesimal model. So each one of these dots basically is like the Q on Ben's diagram that was interspersed with a whole lot of markers. These QTL, these polymorphisms, determine some trait that you're ultimately interested in here. And I've pointed those directly to the endpoint trait, yield in this case. Ben talked about examples from other species. So we're interested in grain yield of our hybrid crop. And we've refined our understanding of which genes we're interested in as regions of the genome, how they interact with each other and use those as models, and then ultimately how they influence traits that may underpin yield, such as the traits that are important in water-limited environments and non-water environments. And within the Center of Excellence, we're trying to take these ideas further, working with some of the detailed biology underpinning some of these traits, such as the work that Christine Beveridge and her team have been doing to understand hormonal regulation of plant growth and development, plant architecture, that have an influence on some of these traits, water use patterns that ultimately influence yield. So embedding this understanding of biology into this prediction framework is what we're actually trying to do. So that's sort of, basically that's my talk. That's the bit that's a bit different from what Ben introduced. And now I'll just quickly go through an example of revisiting the breeder's equation with these principles embedded as a dumb breeder. I'm just a dumb maize breeder now, and I'm trying to operationalize all this so that I can make these decisions, because the data is coming to me and I have to make decisions overnight to send seed off somewhere else in the world for the next stage of a breeding program. So the problem definition, I want to sort of give you a bit of perspective on this, because I think it's really interesting to understand that ultimately, we're not just about selecting for yield and endpoint traits. They come at a cost. So when you have yield from a maize crop, it uses water. So I was really interested in the trade-off of water use patterns around the world for the types of crops we were using in agriculture. And so the breeder's equation can be modified within this framework to help you to get these types of questions. And that underpins a lot of what we were trying to do within this prediction-based crop improvement strategy, and sort of trying to improve the integration of this biological knowledge within the overall breeder's equation. And then start to think about using that information, as Ben also discussed, for designing and modeling the plant breeding program. So for me, there's an empirical world, there's also a modeling world and simulation that I'm doing interactively all the time. So I mean, I spent most of my time simulating all of the scenarios that I was going to try inside my breeding program before I actually did them, using all of the data that we were generating from all the different stages of my program and the 50 other breeders that I was working with inside the company. And we're all sharing that information. And then, so what I'll do is I'll sort of try and go through that theory and embed it inside some of the maize breeding work. So quickly, corn, it's a big crop. So maize, if you've got some idea of sort of the size of it in US agriculture. So for me, US agriculture is a model system that I'm really interested in sort of exploring. It's data rich, and you can try out a lot of these ideas before you start trying them for cassava in Africa. So that's sort of one of the programs that I've also had the chance to get involved with. So maize, just to give you an idea of how big it is, US maize is grown on about something like 35 to 40 million hectares. Might not mean much to people, but Australian wheat's about 10 to 14 million hectares. So we're actually growing something that's much bigger than wheat for the whole of Australia. And the types of hybrids we're producing could be like the amount of wheat that you actually grow in the whole state of Queensland, for example, just as an example of the size of the effort. Maize in the US has undergone tremendous yield improvements over time. And as Ben already showed you from some of the work that we've published, we've looked at the genetic contributions and how genetic improvement has underpinned this in combination with improvement in agronomic management techniques. But as I said, it doesn't come for free. There's no free lunch. You actually use water. And so what I'm just zooming in on here is an area where I was breeding. And this is sort of really nice work done with the GRACE satellites that's actually just monitoring the depletion of the aquifers in the US. So we were actually improving maize productivity, but we're depleting aquifers. And that amount of water use from that agricultural system is significant enough that you can detect shifts in gravity over the agricultural system. So that's sort of the scale. And so what we're actually looking at is the balance of value. So how much is a millimeter of water worth? And sort of how do we actually optimize agriculture for a more circular system rather than just sort of driving for higher productivity? And there's a really interesting recent report that sort of puts some context around this from the Rockefeller Foundation that sort of quantifies the value proposition of US agriculture as it's operating today. So annually, it produces about $1 trillion a year worth of value, agriculture, and corn's a big part of that. But it actually generates about $2 trillion a year of externalities. And part of that is overuse of water and environmental degradation. So what we are actually looking for are breeding strategies that are more in line with circular agricultural systems where we're regenerating the agricultural system. So when I was breeding drought-tolerant maize, it wasn't just about using more yield and producing more yield. Sometimes it was more important to reduce the amount of water that was actually being used and open up whole new water management strategies for the farmers. So just to put that into context. So this is another view of the breeder's equation. And the only real difference here between what Ben actually showed you is I've given some pictures of what's actually happening. If you go from an infinitesimal model with these effects that Ben was talking about for each of these loci that we're putting pressure on, and what we're actually doing with the breeder's equation is predicting the increment from any one cycle of a breeding program. And then you increment that over time to get this long-term genetic improvement that we're continually trying to build on. And this is the underlying shifts in sort of the variation that's happening at each of these underlying nodes that are influencing these traits. A lot of these ideas go right back into the 1930s, where we've developed and refined these prediction methods right back from those early stages. And it's the selection strategies that Ben already introduced to you. And this is sorting out all of the individuals that we're measuring in these experiments or on the genotypic information that we've generated. And underpinning this have been all of the improvements that we've made in understanding the structure and organization of the crop genome, so maize in this case. In the 90s into the 2000s, we started to build reference genomes that we would then use to look at the genetic variation for all these regions of the genome so that we could actually start to understand where we were getting the variability, what structural components, and how that information actually was shared across crops in this case. So this underpinned our ability to be able to create these molecular marker polymorphisms that Ben talked about, the SNPs. What we would do is we would actually organize these SNPs into these chunks that Ben talked about, the haplotype blocks from the founders and how they were broken up and passed from generation to generation. Sometimes we broke them up and then found that we had to rebuild them as we started to understand some of the interactions that were going on with the DNA sequence inside some of these regions. So now you've got this large breeding program generates a lot of the phenotypic data. We got the sequence polymorphisms. We've got this genetic model. We're trying to estimate these effects. So we can actually then do exactly what Ben said, construct these training data sets, estimate these effects. And then if I've got another 1 million individuals that have been genotyped, I can take these effects and I can start to predict my expectations of their performance in other environments, which is what we do. And in the same way that Ben talked about, we explored going beyond the infinitesimal model to start to look at how these regions would interact, and also how we were discovering that over cycles of the breeding program, so that we could translate that knowledge not just into endpoint traits, but how those traits work together and across environments. Basically, this is a cartoon version of this, but we're building response surfaces or landscapes that the breeding program's actually starting to explore. And then with this information, as Ben discussed, we could look at what we're currently achieving, and we could take any one of these scenarios with this information to start to pressure test that and say, on average, could you do better than what we're already doing? And that's basically how we would test out any use of this information for a commercial breeding program. This is on average over many, many different genetic models of trait architecture. And so here, what we've just looked at is different genetic architectures of traits from different levels of complexity. Ben talked about this here. This is low level of complexity to high levels of complexity with more epistasis, more G by E interaction. And what we actually learned very early on was, as we built these models, that what Ben showed you empirically was that we could get more value if we could get appropriate models for the more complex traits, and then use that information to prime our predictive selection strategies. So that's ultimately why we were investing in a lot of algorithmic development to try and build a knowledge of the underlying architecture of these traits, so we could start to ask questions about any one breeding strategy. Where is it taking you? Can you actually design a breeding strategy to avoid it going here when ultimately you want to actually go here? And that's the sort of balance between yield and water, for example. This may be a strategy that gets you to a high yield, but uses a lot of water. This may be the strategy you might ultimately want to get to with equivalent yield, but less use of water, for example. And so this sort of takes me back to this overall sort of progression that I've talked about. Sort of the cartoon version of this is from the infinitesimal to trying to use this biological information in these intermediate layers so that we can actually better predict some of these endpoint traits. And within the Center of Excellence, while we've done a lot of this in maize in the past, we're now trying to translate it into other crops. One of the crops of focus for us is grain sorghum, which is a major summer crop for us in Australia. And so working with David Jordan as the breeder and a bunch of others, Christine here, on trying to look at the transferability of this to other target crops. So sorghum as an example here. And so this is just to remind me, this green box, that I looked at Scott's video. Scott told you all that I was going to explain a slide he showed that he said he wasn't even going to try and explain. And I always do what Scott tells me to do. So this is the slide that he actually put up, and he said I should explain this, and he wasn't going to try to, so I'll try. So this is sort of the algorithmic model that underlies implementation of using something like a model where you have knowledge of how traits interact to influence yield, to actually build a better prediction of the yield outcomes. So instead of trying to predict directly to the yield outcome, what we're trying to do is we're trying to use our knowledge of the variation of some of these traits that are influential on our outcomes in combination with these crop models to actually supervise our search algorithms with the crop models to predict those traits, and then use the crop model to actually navigate the landscape to the yield outcome. Okay, so there's no magic. It's just we're using the genetic information in combination with the biological model, and we've actually embedded that right inside the predictive algorithm. So these methods are being used now to build maize hybrids for the US Corn Belt. We would like to do this for a lot of other crops, for a lot of other situations around the world. And so then a lot of these algorithms that Scott talked about for phenotyping in his talk, we're using them to think about how would you build prior knowledge and use that to actually parameterize these types of predictive models to embed that biological information into these predictive algorithms. And ultimately, what we'd like to be able to do, and what beyond being able to do this for maize is to use the predictive outcomes from these processes to be able to distinguish more easily between this type of hybrid, where we're looking at a continuum of water availability from high water availability, high evapotranspiration in this case is the way we're quantifying that, to a drought environment with low evapotranspiration outcomes. Here's a hybrid that actually sustained yield over that in that range of environments in this experiment, and then another hybrid, which was another commercial hybrid that's grown on millions of acres in the US. Growing that across those environments, you can see that it ran a very high risk if it was caught by drought. And so this then building strategies to search through all of that genetic space for these types of environments has allowed us to move over multiple cycles of programs to try and reduce the gap between what's potentially possible, what we're currently achieving. So we're actually trying to put selection not just on yield, but on the yield gap between what's possible and what we're currently achieving, so we can actually reduce that yield gap. And this yield gap that I'm quantifying here is not just over cycles of breeding, but this is the cycles of breeding after the farmers have decided to adopt these hybrids over millions of acres in the Corn Belt. And so what we've actually done is we've, over a couple of decades, has greatly reduced this yield gap from on-farm productivity. So we can actually look at the improvements in yield outcomes without overusing or reducing, in some cases, the amount of water that's actually being used to achieve that yield. So we can actually get sometimes more yield using less water in this case. And so the intersection and opportunities, just really quickly, I think totally interested in anybody's and everybody's ideas about what are the best ways of advancing these multi-trait gene to phenotype models for traits. And also what sort of prediction methods, just exactly as Ben said, on ways of refining and improving these search algorithms. And similar, over a couple of decades, pretty much I found the same thing that Ben talked about, that unsupervised algorithms basically went off on tangents that I wasn't really interested in. So we're actually using these biologically inspired search algorithms basically as supervised search algorithms to try and focus on the components of this highly multidimensional space that we want to actually explore and exploit. And so that then sort of puts us in a position where you can actually think about a breeding program as a very empirical thing, but you can also think about it as a biologically inspired search algorithm. And we're interested in sort of how we can improve on that. And we're always looking at ways of improving the simulation methods that actually go with that. And just to quickly say that this is obviously done as a great partnership with other CIs inside the Center of Excellence. So this is the work beyond maize, but looking at doing this for other crops. Of course, looking at the traits, the mechanisms, the modeling and prediction methods, and how you would actually put those into practice for agricultural systems. And again, the Center of Excellence isn't trying to do this itself. It's doing it, working on these prediction methods in partnership with a lot of partners that are on the ground in these agricultural systems, trying to deliver the outcomes to farmers. So I'll leave it there. Thank you very much for your attention.
Okay, we'll have the Q&A session now. So just come to the different view, presenter view. Stop share. There we go. So we might start with some of the questions. We'll give Mark and Ben a mic from online. So there's some questions here. Looks like the first question maybe to Ben. This is probably, yeah, it's an interesting one. Is there any open sources for fast stack paper or codes? Like, if you want to do this at home, can you do it at home, Ben?
Yes, the good news is you can. I think it's up on GitHub. We're just writing, or Kira's writing more precisely, the paper, and she's about to submit it. but it is up on GitHub, the code. I just have to confirm that with Kira. I can't see it. But if you're interested, just send me an email and I'll send you the GitHub link.
And another question for you, Ben, on the machine learning models for fiber sugar yield prediction was about 50% max from the data you were showing, the results. How does this compare to traditional approaches for that type of trait in sugarcane breeding?
Yeah, it's not too bad. You'd need a reasonable number of plots if you were just trying to get to that accuracy by traditional phenotype selection. I guess the point is you can get this breeding value or this information much earlier. So you could get those predictions from a seedling rather than having to wait till you actually have plots and actually measure it across a large number of plants. So the phenotypes are probably more accurate once you get to that point, but you have to wait a long time to get to that. Whereas with the genomic predictions, you can breed from those individuals earlier in the cycle.
This is an interesting question around recombination. Does your genetic algorithm consider random recombination, or is there any systematic downstream principle for recombination when you're generating your offspring? How do you sort of take this into account?
So we've done that in two ways. We have a random recombination operator, and we're also building in a hotspot of recombination model, if you like, based on where the historical recombinations in crops like wheat have actually been observed. And you can do the same thing in cattle and humans. There's definitely hotspots of recombination across the genome, and we're building that into the code. I don't think it's in the first version on GitHub, but it's on the way.
Oh, this is a strange person, Kai Vosfels. He says, great talks to both of you. Ben, have you looked at including any additional sources of information in your machine learning algorithms that you use for prediction? Example, weather data.
We haven't in the machine learning approaches here. We have done that in cattle data to start to partition out things like G by E that Mark was talking about in I guess, a little bit of a crude way. And you can definitely see some G by E interaction when you do that. So it's a good point, but we haven't tried it in our machine learning models. One of the next things I'd like to throw these models at is these G by E problems. And there you could definitely start to incorporate weather data. There's a lot of it. It's not quite free, but pretty much from the Bureau of Meteorology. So it's a good source of data.
And he was also wondering, he's pretty demanding. He's very fussy German. He wants to know how far we can push these prediction accuracies. Are we at the limit, or can we go further?
So interesting question. In cattle, where some countries are up to about 1 million animals that have phenotypes and genotypes recorded, they get an accuracy for something like milk production of about 85%. And it hasn't improved since they had about 100,000 records. So it doesn't seem to be capping out at 1 for some reason that we don't really understand. I guess non-additive variation, different sources of variation would be a key culprit, but we certainly need to work on that. I believe the human geneticists, with their absolutely massive data sets of like 5 million people and full genome sequence, are now up in the order of 80% of the variation captured. But again, it seems to be plateauing, and we don't really know whether it's a true plateau, whether it's going up very, very, very slowly, but there's certainly a bit of a gap left. 85% is incredibly useful as a genomic predictor if you have that high accuracy, but of course we'd like it to be 1. So good question.
Thanks, Ben. Well, a question for Mark, also coming from Germany. Which infrastructures are needed to put into place to train the next generation of commercial breeders that will be able to implement those highly complex transdisciplinary and cross-scale modeling approaches that you've outlined, Mark? And do we need more dedicated training centers or grad schools, et cetera, to sort of breed the next generation of breeders?
I don't know about breeding the next generation of breeders, but anyway, so on the integrating the biological capabilities in with the genomic prediction, this is one of the targeted outcomes that we have for the Center of Excellence, is to actually build that as a platform by merging the prediction capabilities with the AppSim crop growth model that is actually developed by the teams based. I mean, one of the key teams, Graham Hammer, led that for the development of a lot of the crop modeling capabilities. So instead of having to build your own crop model, you'd actually access that platform in combination with these prediction capabilities, and then you could look at how you would improve the model and use the biological information for your purposes. So one of the outcomes that we're aiming for is to actually enable accessibility to these capabilities by building that platform and working with the partners to make that an open source opportunity for people. And so that sort of is what we're working towards within the Center of Excellence. In terms of training, there's no doubt that there's ways in which we can improve the closure of the gap between the training that new graduates can get today in sort of a very specific focused project and the way that large scale commercial breeding programs operate today. There's a huge gap. And sort of having been involved in training on both sides of that, I mean, within the multinational companies, they're looking at how to work with universities to help close that gap, and thinking about how to do training differently than it's been done in the past with more interaction is really important. I think that's also equally important for these coordinated efforts for these crops that people often refer to as orphan crops that don't have the same critical mass of infrastructure underneath them, and thinking about the transferability of these capabilities. And so I think that there is a lot of discussion on how do we put in place the training that's necessary for that. And I know that some of the relevant people involved in those discussions are in this room.
Well, thanks a lot, everyone. I think we might stop the questions there. I know there's like just hundreds of people in this room wanting to ask Mark and Ben more questions, but to get you to interact, I think that's a great excuse to do that over some food and drinks outside. So we might stop there. Thank you everyone for joining online and in person. So please join me in thanking our speakers today, Mark Cooper and Ben Hayes.